GeneX is a unique biphasic bone graft which possesses a controlled, reproducible negative surface charge. It is hydrophilic, has a physiologic pH, and is fully resorbed and replaced by bone. These properties make GeneX a huge leap forward in bone graft engineering. All materials, when placed into an aqueous solution, can exhibit a surface electrical charge. This results from the interaction between the surface and the ionic species in solution. This charge is known as the zeta potential, a basic law of nature playing a crucial role in biological systems. Cells in the body normally have a negative surface charge. Proteins in the body also have a surface electrical charge that varies with the protein and its function. GeneX contains a surplus of electrons that, when implanted, presents a controlled and reproducible negative surface charge relative to the surrounding biological medium. The protein-rich extracellular matrix surrounding the cells possesses an overall positive charge, and key species are attracted to GeneX because of the negative surface charge. Aided by its hydrophilic nature, GeneX is rapidly coated in this protein layer. The adsorption of proteins on the surface of an implanted material translates the structure and composition of the surface into a biochemical language to which the cells can respond. Osteoprogenitor cells and mesenchymal stem cells detect the presence of the osteogenic and osteoinductive proteins, triggering the proliferation and differentiation of the cells into bone-forming osteoblasts and driving attachment of the cells to the surface. This layer of osteoblasts covers the graft material, laying down new bone. As the osteoblasts mature in the bone matrix, becoming osteocytes, the front line of osteoblasts is replenished by the further proliferating osteoprogenitor cells. The calcium sulfate and beta-tricalcium phosphate constituents of GeneX create a two-stage resorption profile and provide high local concentrations of calcium and phosphate ions, which reprecipitate onto the surface as a bone-like apatite layer, further stimulating the bone formation cascade. This layer encourages the mineralization of the newly forming bone and initiates coupled osteoclastic resorption. The high-purity calcium sulfate resorbs first, creating a porosity ideal for vascularization and early bone ingrowth. The slower resorbing beta-tricalcium phosphate provides an ideal scaffold for new bone formation and is removed by osteoclasts in harmony with new bone growth. The newly formed woven bone is eventually remodeled into mature lamellar bone, completing the bone-forming process.